All right, so welcome to the second video in our photosynthesis lecture. This is actually going to be our final video. We're really just going to talk about the process of photosynthesis and how it actually works at the cellular level. And we can divide that process into two different stages. We can divide it into the first stage, which is the light reactions. This is where the chlorophyll in that thylakoid membrane actually absorbs that solar energy and converts that solar energy into chemical energy, such as ATP and NADPH molecules. And once those ATP and NADPH molecules are made, they then move on to the light independent reactions, or the Calvin cycle, which is where the energy from those molecules are made to fix carbon into a sugar. So to fix carbon from carbon dioxide into glucose, and then glucose can be used to make cellulose or starch in the plant. So remember, starch is the energy storage molecule in plants, and cellulose is kind of that building block molecule in plants, helps keep its structure. And so here's just kind of a roadmap for photosynthesis. So basically you have your chloroplast, you have your water, you have your light, and so that is used to make your ATP and NADPH molecules. That gives off our oxygen. Then those molecules, those energy molecules, go into the Calvin cycle, and then they take the CO2 and turn it into sugar or a glucose molecule that then can be used for, say, building of starch or cellulose, and can be used in any other cellular processes that they need. So the initial incorporation of carbon from the atmosphere into organic compounds is what we call carbon fixation. So we're basically taking that atmospheric carbon and converting it into an organic molecule. So the plants are fixing that carbon. And this can actually help reduce the concentration of carbon in the atmosphere if you have enough plants, enough autotrophs taking it up across the globe. And so the relationship between photosynthesis and carbon fixation is a good example of how all of life is kind of interacted and is interconnected with one another. So that's one of the big components kind of from our first chapter. And so if we think about this little question here, so what molecules are the inputs of photosynthesis? What molecules are the outputs? So take a minute just to refresh your memory. All right, and so the inputs should be carbon dioxide and water. Outputs should be glucose and oxygen. Okay, so we said that the first process in photosynthesis, or the first step, is the light reactions. And so the light reactions is where the plant takes the solar energy and converts it into the chemical energy. So the ATP and the NADPH molecules, okay? And it gets that energy to put into those molecules from the sunlight. So the photons that are given off by the sunlight. And solar radiation travels through space as rhythmic waves, okay? And so it's important to understand what these waves actually are and what's actually going on in order to understand kind of the light reaction process in photosynthesis, okay? And so as solar radiation is traveling through space as these waves, you can actually think about them as like waves on the ocean. So it has a crest and a trough. And so we can look at the distance between two adjacent crests of a wave, and you can classify that as the wavelength. So the wavelength is basically the distance between two adjacent crests. And if you look at all of the wavelengths, so the full range of radiation, the full range of wavelengths, is called the electromagnetic spectrum. So here is a figure showing the electromagnetic spectrum. with smaller wavelengths on the left-hand side, so those crests are closer together, to larger wavelengths where the crests are farther apart. So for example, radio waves have wavelengths that are 10 to the third meters apart, so very far apart, whereas gamma rays have really, really small distances. We're talking about 10 to the -5 nanometers apart, so very, very small. Now, the visible spectrum, so the visible radiation that we can see as color, is from 380 nanometers to 750 nanometers. And depending on the wavelength determines the color. So for example, shorter wavelengths are purples and blues. Longer wavelengths are reds and oranges. So here we're showing a wavelength of 580 nanometers. So that's the distance from this crest to this crest. And that is this orange looking color here. Okay. And so the wavelengths are important because it determines how much energy is in that wavelength. And so the way that scientists kind of determined what wavelength was being used in photosynthesis is through the work of a pretty savvy biologist, a German biologist. So in 1883, this biologist, Theodor Engelmann, basically noticed that there were bacteria that was living in water, and they lived in clusters where there was higher oxygen concentrations. And so he basically said, okay, these are oxygen-seeking bacteria, and so they will congregate near areas of algae that are performing the most photosynthesis. So if they're doing the most photosynthesis, they're releasing the most amount of oxygen. So that's where they will congregate. And so what he did is he took an algae cell and he exposed it to different wavelengths of light in the visible spectrum. So from 380 to, I think it's like 740, right? And he placed that algae on a microscope slide and basically observed where the bacteria cells would congregate. And what he saw is that most bacteria congregated around the red, orange, and the blue violet wavelengths. So basically he let the light go through a prism and separate it. That prism separates it into its different wavelengths, and then he can observe that on the algae cell. And so basically this portion of the algae cell, which is where the green yellow wavelengths are, is not doing much photosynthesis because there's not really any oxygen here because no bacteria is congregating there. Whereas the most bacteria is around, 420, 430 wavelength and 700. And so that means that's the wavelengths that are kind of being prioritized in the process of photosynthesis. And so what that means is that the chlorophyll pigments are absorbing this wavelength and reflecting that green wavelength. And so again, that's why we see plants as green. That's why we see chlorophyll as green, because that is a wavelength they're reflecting. And so those pigments that are in the chloroplast, so for example, the chlorophyll, they are selective in what wavelengths they absorb. And so that light color that's poorly absorbed by the chloroplast and reflected is that green color. So that's green wavelength. And, this just keeps in mind the fact that energy can't be destroyed. So it's absorbed or converted into other forms. And so that energy is absorbed by the chloroplast and then converted into that chemical energy. And so again, that reflective green light is what we're seeing. So any object you see, the color is the color that the wavelength that's being reflected, and all the other wavelengths would be absorbed. And so chloroplasts have a couple of different pigments that absorb different wavelengths of light. So we mentioned chlorophyll already, but there are chlorophyll A and chlorophyll B pigments. So chlorophyll A mainly absorbs the blue, violet, and the red light. Chlorophyll B is not really in the light reactions, but it conveys absorbed energy to chlorophyll A. And then the light reactions will continue from the energy in the chlorophyll A molecule. Chloroplasts also have these family of pigments called carotenoids. These are kind of yellow-orange pigments, and they mainly absorb blue-green light. And so having a vast array of these different pigments that are absorbing different wavelengths of light really leads to the variety of colorations that we see in plants, and it's reflective of the wavelengths that are reflected in that process. All right, so now that we know how light works, how solar radiation travels as these wavelengths, and how there are specific pigments that are absorbing those specific wavelengths, now we have to figure out or explain how the molecules in the chloroplast organelle convert that solar radiation into chemical energy. So basically, that light travels as a wave, and it's basically in these packets called photons. So little waves that are referred to as photons that have fixed specific quantities of energy. So again, the shorter the wavelength, the more energy that photon or that packet of light has. So what happens is you'll get a pigment molecule that will absorb a photon, and then that energy from the photon will basically excite an electron, and that electron will gain energy. And as that electron loses energy, it falls back down to what we call a ground state. And so most of these pigments will release their heat energy as their electrons fall back to that ground state. And also some pigments that are excited will also emit a light as they fall back down to their ground state. And these are known as fluorescent lights. And so, for example, the light that you see in a glow stick is caused by the chemical reaction that's exciting the electrons and the fluorescent dye. And those excited electrons will fall back down to the ground state, releasing that energy. And that's why the glow sticks don't last forever, because that energy will eventually be released. So here you have a figure of that, where you have the light hitting that chlorophyll molecule. The energy in that photon is exciting that electron. As that electron falls back down, it releases energy, and it releases the light fluorescence that leads to the glow sticks that we see in this picture here. Okay, so the chlorophyll molecules are located in the thylakoid membrane. So remember that chloroplast has the granum, and the granum's made up of the thylakoid membrane stacks. So inside of those thylakoid membranes, there are clusters of pigment molecules that are in what are called photosystems. And each photosystem has several hundred pigments of chlorophyll A, chlorophyll B, carotenoids, and all that depends on the species of plant that the photosystem belongs to. And that photosystem and the cluster of pigments that it holds basically functions as a light gathering antenna. So it gathers all the light that comes into contact with that chlorophyll molecule. So here we have a figure showing this, where we have a chloroplast, we have granum stacks, grana stacks with thylakoids. And if we zoom in to one thylakoid membrane, here's that phospholipid membrane with the pigment molecules inside of that photosystem. So this green object is the photosystem, and those little circles on top of that are the different pigment molecules. So it's chlorophyll A, chlorophyll B, etc. And so basically what happens is there's electrons here. The photon packet will hit that electron, cause it to become excited, and then it will fall back down, and then another photon will hit, and it will continue to travel across those chlorophyll molecules. So this is called the reaction center, right? And these pigment molecules is transferring that photon energy into the electron. Okay? And in total, for the light reactions, there are 2 photosystems that work together. Okay? The photons are entering that photosystem and interacting with the chlorophyll and the electrons that are on that chlorophyll. And so the photons excite the electron in that first photosystem. That energized electron will then travel through several different proteins, and this is called the electron transport chain. And so as that electron travels through those proteins, it uses that energy that's being released as it moves back down to ground state to make ATP. Okay, and so the electron transport chain, the ETC, gathers energy from that electron to make ATP. Then that electron will be at the second photosystem, and the electron will have a photon to hit it, will become excited, and then that energy will be used to generate NADPH, so a molecule of NADPH from NADP+. So here we have a figure showing the process of this light reaction. So you have water, and water is necessary here because this is where the electrons come from. So as the light hits the chlorophyll molecule, the electron is excited. And as it falls back down to ground state, it travels through those different proteins that make ATP. Okay? And as this electron is excited, another water molecule will be split, and the electron from it will take its place. Okay? And so this splitting of this water molecule is essentially breaking it apart. So you have oxygen molecules leaving. That's where the oxygen gas comes from in the photosynthesis process. Okay. So once this electron makes it to the second photosystem, it then is hit by another photon packet, and that electron is re-excited. And it is then used to turn NADP+ into NADPH. Okay, so that first photosystem makes ATP with the energy from the electron in the transport chain, and the second photosystem makes NADPH from the energy in the electron after it's excited again. Now, the ATP and the NADPH molecules will then move on to the Calvin cycle, or the light independent reactions, to fix carbon into glucose. So to fix the carbon from carbon dioxide into glucose. Okay, and so those light reactions are located in the thylakoid membrane, and the 2 photosystems and the electron transport chain transfer electrons from that water molecule to make ATP and NADPH. Okay, and so basically we have another figure here showing this process. You can see photosystem 1, photosystem 2, the electron transport chain. You can see water being split, and water is required because it needs to donate that electron. If you don't have any electrons, you don't have any electrons to get excited, so that energy can be transported through that electron transport chain and make ATP, and then ultimately make NADPH. Now, the way ATP is made is as that electron travels down that protein chain, that electron transport chain, the energy from the electron as it passes through a protein is used to actively transport hydrogen molecules inside the thylakoid membrane. So what the membrane or the chloroplast is actually doing is setting up a gradient. So it's using energy to set up a gradient of high hydrogen ions inside versus low concentration outside. So that means that hydrogen ions want to diffuse from inside of the membrane outwards. So they would passively diffuse from the inside to the outside, but they're actively brought in from the outside to the inside via the energy in that electron transport chain. So its electron transport chain is setting up this gradient because there's an ATP synthase molecule in the membrane that the hydrogen ions will passively move out of. And as they flow through that enzyme, it's basically like water turning a turbine, that energy will be captured and transported into the ATP molecule. So that energy is used to take an ADP molecule and attach its third phosphate group to it to make ATP. And so that's how the ATP molecule is made, is through the setup of that hydrogen proton gradient with the electron transport chain. So here's another example of this light-dependent reaction. So what we have here is the men with hammers actually serving as the energy from a photon. So the hammer hitting the seesaw, adds energy to the electron and excites it. And then it will kind of go down the transport chain, lose energy. Then you'll have another hammer hit it and it will be re-excited. And then that energy will be made to make NADPH. Now, remember, these two molecules, ATP and NADPH, move on to the light independent reactions, the Calvin cycle. Okay. And so one thing I just want to note is that the mechanism of ATP production is very similar to that in cellular respiration. And so that electron transport chain pumps the hydrogen ions across the membrane, and then that ATP synthase enzyme uses that gradient to make ATP. The main difference, though, is that the food provides the high energy electrons in cellular respiration, whereas light is providing the energy for photosynthesis. So it's those organic molecules, glucose, providing energy in cellular respiration, sunlight for photosynthesis. And so once that ATP and NADPH moves on to the Calvin cycle, this is basically where that glucose molecule will be made. So the Calvin cycle will take a sugar molecule called G3P, and will take carbon from the atmosphere and energy from ATP and NADPH, and will convert that into basically a 6-carbon molecule that will be broken down into a 3-carbon molecule, the G3P sugar, and then that G3P can be used to make glucose or other organic compounds. So let's just look at a figure of this, okay? So here we have, so here we have your carbon dioxide from our air, right, from our atmosphere. We have what's called an RUBP sugar, and this sugar has 5 carbons, right? This carbon from CO2 will be attached to this molecule, and it will turn it into a 6-carbon molecule. And there's an enzyme called Rubisco, R-U-B-I-S-C-O. You don't need to memorize this, but just letting you know, Rubisco is the most abundant enzyme on the planet. Rubisco will take the carbon from CO2 and fix it onto this 5-carbon molecule. Now that is a 6-carbon molecule. That 6-carbon molecule will be broken down into this 3-carbon molecule, and then using the energy from the ATP molecule made from the light-dependent reactions and the NADPH made from the light-dependent reactions, this molecule is converted into a G3P sugar, okay? Now, if I remember the numbers, I think it's 12 of these G3P sugars are made. 6 of those, or I'm sorry, 10 of those will go on to be recycled into an RUBP sugar. 2 of those will go on to make glucose. So these G3P sugars are 3 carbon molecules. So remember, glucose is 6 carbon. 1, 2, 3, 4, 5, 6. Okay. And so you'll have 2 of these molecules to make a glucose molecule, or they can go on to make other inorganic compounds. So basically what you would need to be able to summarize from this Calvin cycle is that you have CO2 from air being fixed into an organic molecule. That organic molecule is then broken down using ATP and NADPH and modified into a 3 carbon sugar, that G3P sugar. And then that G3P sugar, there's many of these made. Some of those will go on to make glucose. The other will be recycled to be used again in the Calvin cycle. And so then that glucose molecule can then be made into starch or cellulose to become a constructional component for the plant, or to be an energy component for the plant. And so that's pretty much what the Calvin cycle is. Okay, and so that's it for our video for photosynthesis. So we went through a basic overview of photosynthesis, you know, why it's important, why we need to understand it, why it's important for ecosystems and other organisms, and then the basic processes that happen at the cellular level for those 2 different parts of the photosynthetic process. So the light-dependent reactions and the Calvin cycle, which is also known as the light-independent reactions. I should state it's known as the light-independent reactions because the Calvin cycle does not need light in order to work. It can work at daytime or nighttime as long as it has carbon dioxide and ATP and NADPH molecules. Okay, and so with that, I hope you all have a good rest of your module. Make sure you complete all of your assignments, and we will see you in the next module.